So welcome to our March coffee hour. My name is Agnieszka Carpenter. I'm the Executive Director of BioMe. BioMe is Bioscience Association of Maine, and we are a trade industry group devoted to advancing bioscience in the state of Maine and beyond. Virtual Coffee Hour is our monthly series where we highlight 4 members of our bioscience community in Maine who talk about some exciting developments, new companies, entrepreneurial projects, and research that's happening in our state. And it's sort of a replacement of networking in COVID times, but I think we will continue with this virtual series when the pandemic ends because it's a very popular event for us. And I wanted to start by introducing our speakers. So today we will have Kate Dickerson, founder and director of Maine Science Festival. We have Dr. Anne Lichtenwalner from UMaine, who's the associate professor of animal and veterinary sciences. We have Dr. Matthew Lynes, faculty scientist at MMCRI, and also Theresa Roelke, a nurse practitioner from MaineHealth, who has a cool technology to share with us today. I also wanted to thank our sponsors. These are our BioMe annual sponsors, and we would like to thank you for your continuous support. We're able to do these events thanks to the support that we're receiving. A couple of logistics before we dive into our presentations. The chat function is on, and we ask that you use the chat function. You can leave your questions in there for our speakers because we will have a 10 to 15 minute Q&A at the end. If you've attended any of our coffee hours in the past and you're familiar with that whole setup, you can also use the chat to just say hi or introduce yourselves or comment. We welcome any comments from you. Okay, with that, I will stop my share. And I would like to introduce our first speaker, and it's Theresa Roelke from MaineHealth. Theresa, you can share your screen whenever you're ready.
Okay. Here we are. Can you all see?
Yes, thank you.
Okay. So thank you for having me. I am a geriatric nurse practitioner. Prior role was thoracic oncology, where I manage the lung screening program. My current role is within the Maine Medical Center Research Institute, where I work with the long COVID research, and I also work as a provider in the long COVID clinic. So statistics that'll make you pause. Roughly about half of the CT scans in adults will show one pulmonary nodule within a lifetime. Solitary pulmonary nodules are one of the most common thoracic imaging abnormalities that we find. Each year, about 1 million nodules are discovered as incidental findings. And within the context of lung screening, the prevalence of solitary pulmonary nodules ranges from anywhere from 8 to 51%. So what's the problem? Well, each year we've got tens of thousands of Americans and really people around the world who will learn that they've got one or more lung nodules on low-dose CT imaging within the context of lung screening, or as an incidental finding on chest CT imaging. So oftentimes the patients won't understand what this means to their overall health. They'll be left with distress and some degree of anxiety. And then added to this within the US is our lack of understanding of metrics within the society. And so a lot of times people will think that a 3 millimeter nodule is a lot larger than it is. And one of the first questions that they ask is it cancer? That's what they want to know. And so the story of pocket nodules actually begins literally within a clinic, within the thoracic oncology clinic, within actually a closet that was being cleaned out by a nurse for a different use. And so she found this resin nodule tool, and she brought it to me and said, I think you might be interested in using it with your patients. So I brought it home, I measured the nodules with calipers, and I began to use it in clinic. So if you look over on the bottom right, you'll see the actual 3D nodule tool that's currently in use. We are revising that design at the moment. But the tool is really designed to help patients understand pulmonary nodules wherever nodules need explaining. So this could be within the context of oncology, within lung screening program in primary care, within pulmonary clinics, and then also within emergency departments. So this is a slide just showing that this project started about 4 years ago, and I began working with a student from Mecca to prototype the tool. And so if you look at the top, you will see that the student over here, William, on the left, and then over on the far right, you'll see the first prototype. And the fun part about that is he actually printed this with a 3D printer that he got for graduation from art school. If you look at the bottom, this bottom left, you'll see the first resin tool that was actually found in the clinic. And then it takes you through the various stages of prototyping. So who benefits from the tool? Well, patients and also clinicians, because it makes life easier to explain pulmonary nodules with a visual, because it creates experiential learning for the patient, and it's more likely to stick when you create an experience with patients. So I did a 31 question patient survey, and actually with 31 patients, I did a short survey, and 97% of them found the tool very helpful in explaining lung nodules overall. And I asked about, how did they feel about understanding size, characteristics of a lung nodule, and so on. And many found it very, very helpful. So this slide just shows you the enthusiasm that patients had for the tool, and also clinician enthusiasm. And this has been ongoing, this enthusiasm, especially among clinicians. And, they often will ask, when are these going to be available for use? How do I get one? So the market is pretty broad. As I had mentioned earlier, it can be used in oncology, hospitals, emergency departments, in many, many settings, not only across Maine, but across New England and really across the country. And so the goal is to really, again, create that experiential learning. I'm forming an LLC called Teach Me RX, and we're starting with the pocket nodules lung. And then the idea is that if that all works well, then we're going to extend the brand to include nodules for other type, tools for other type of nodules that are found. And you can see that shown here. This is a manufacturer I'm working with from Kennebunk called G&G Manufacturing, a local manufacturer. They have been amazing to work with. We are doing the final tweaks to the design to create molds for injection molding so that we can create these things in volume. And the reason for that is because I have interest from the International Association for the Study of Lung Cancer. I had presented the tool at a summit in last May. And as a result, the Lung Ambition Alliance group connected with me and asked if they could co-brand the tool. This resulted in another presentation to the Early Detection Committee with ISLAC. And now I'm working with ISLAC, Lung Ambition Alliance, and AstraZeneca in order to get that tool out there. And we're looking at probably about 2,000 or maybe a little bit more to print these tools in volume and then send them internationally, for use internationally. And the idea is that they have an annual conference, a world conference. And so to really introduce that tool at the conference and then to begin to get them out there further. And so we're working a little bit more on the design modification. I'm working with a couple of physicians you see here, Stephen Lamb and David Yankovics. And we're just designing a short survey for providers to collect a little bit more data on the tool and how they feel it is in terms of using it in clinic and the success of it. So what's missing from lung cancer screening, because I think that our volumes are still pretty darn low. And I think part of it is effective branding. So if you look over on the left, you see the Susan Komen Foundation logo, and you see that pink ribbon is tied to that foundation. I think that ribbon is recognized globally, the pink ribbon. I think people may struggle or often struggle with what other colored cancer ribbons look like, what do they represent? And so I think if we can somehow tie that white ribbon something. I'm thinking pocket nodules, you might, get some more interest, and then also tying it, identifying it, connecting it with lung screening. The other thing that I think is missing is support in helping people quit smoking within the context of lung screening. So I'm working on an app, just really in the early stages, that would help track smoking for patients, their daily smoking, the amount that they will smoke over the course of weeks to months to years, and then also tracking CT results, so low dose CT results within the context of lung screening, but also getting patients to understand their RADS category, the nodule size, finding, location, the date of the scan, and then their follow-up, so that they come back to follow up for their annual screening. And what should be missing from lung screening, in my opinion, the word cancer, because it evokes negative emotion, and people, that's a frightening word for people. It causes a lot of anxiety, as so many know. And so, something that we should consider. And basically, my thinking is to give hope to patients by embracing the current technology, creating a paradigm shift in lung cancer screening, because of the people who are eligible, we're really only capturing about 5 to 6%. And that's it. Thank you for listening.
Thank you so much, Theresa. That was really fascinating. And I do see some questions coming in, so we will answer them at the end. If you have any further questions for Theresa, please leave them in the chat. So now we're moving to our second speaker, and it's Dr. Matt Lynes from MMCRI, who will tell us all about brown fat, which is also a very fascinating subject. Matt, whenever you're ready.
Oops. Good morning, everybody. Thanks for having me. Like Agnieszka said, my name is Matt Lynes, and I'm a new researcher at Maine Medical Center Research Institute. And I'm nervous to start off my talk. I think you're supposed to make the audience like you, but I have to admit to you that I've recently moved to Maine Medical Research Institute from Massachusetts. I know that that's not a great look right now. But it's to further my research. I've started my lab here at MMCRI. I still maintain an appointment at the Joslin Diabetes Center in Boston, Massachusetts. But I want to study adipose tissue and thermogenesis, which I'll tell you about today. And so I'm happy to have this opportunity to tell you about the work that we're starting here at MMCRI. I have to start with disclosures, specifically because they relate to what I'm going to talk about today. I hold patents on this molecule I'm going to tell you about called 1213-dihome. My wife holds some equity, but it's not related to what I'll talk about today. So I study the problem that I want to address is the endemic of obesity and diabetes. So these are some sort of pretty visuals that hopefully convince you what most people accept is true, and that is that obesity and diabetes are becoming more prominent both in the US and globally. And the reason that that's bad, and even down in Maine, diabetes incidence is increasing. And the reason that that's a problem is because obesity is a risk factor for pretty much any disease that you can name, cancers. Even now we know that it increases the risk of COVID and the risk of death from COVID. But really, obesity is associated with an increased risk of all-cause mortality. So if you are obese, you are more likely to die. And that is a human health risk that we want to try to contribute to stopping. And so what's causing this increase? Many different things. There are social influences, economic influences, but in my group, we focus on a very simple concept, and that is that obesity is the result of some sort of imbalance between the energy that we take in and the energy that we expend in our lives. And to focus even more, because there are many organs and tissues and cells that control energy balance. You can imagine your brain telling you to eat, your intestine, and even it's becoming now more apparent that the bacteria that live in our intestine can govern how we extract energy from the food that we eat. But then the organs that I specifically focus on are the fat the energy storage organ, your white adipose tissue. And then, of course, what do we use the energy for? We use it to live, to do our thing, to move around, to do our thing. But now what's becoming more apparent is we also use that energy. And again, it's nice to talk to an audience from Maine who accept this pretty easily. We use it to keep ourselves warm. And it turns out there's a specific tissue in your body that you use to keep yourself warm. And so I focus on, and all these tissues talk to one another, of course. I focus on white and brown adipose tissue and the sort of dichotomy that they have in energy storage versus energy expenditure. And just to not dip too deep into the molecular biology that we study in our lab, we know that the way that brown fat makes heat, keeps you warm, is that it actually changes a fundamental process in all of your cells that is the reason we eat our food. So all of our cells use the food that we eat for metabolism, and they make ATP so you can move around. This is called oxidative phosphorylation. But in our brown fat, and exclusively in our brown fat, there is a mechanism that blocks this. And so in brown fat cells, and only in brown fat cells, no ATP or very tiny amounts of ATP are made for cellular work, except they still participate in this oxidation of glucose and lipids in the mitochondria, except they don't make any work. They don't move. And so what happens? They get hot. And you can regulate this process to activate it when you're cold. And the cool thing is you can see this in people because, again, the brown fat will still oxidize this glucose and lipids. And so thank you to the previous speaker for talking about CT imaging. This is actually PET CT imaging. So this person has been injected with radioactive glucose, exactly like I showed you should go into the mitochondria. And they've put their arms up in the air and we've taken a picture of them. And in black, you can see that radioactive glucose. Their head is cut off, so you can't see it going into the brain. And so then what we ask these people to do, or what this study asked these people to do, is wear this surgeon's cooling vest. And so this will, pump cold water through all of the little blue hoses that you see on the t-shirt. And then when you ask them to do this radioactive glucose CT, you see all these black spots, right? And I put red arrows in case. Yeah, I don't know why I put the red arrows. And so this is brown adipose tissue. This is tissue in there, in between their vertebrae and in between their shoulder blades, sort of, that is taking up glucose in response to the cold exposure. And we know that the amount of brown adipose tissue people have decreases with age and obesity. So having more brown fat seems to give you this younger, less obese profile. But now what we know is that about half of the people have brown fat and half of the people don't. And having it is associated with protection from cardiovascular disease. So that's great. So the last part is that there are very few studies about, well, what can cold do for you? And I feel like, again, if I could see all your faces, you'd all be nodding. What can cold do for you? I can tell you. But we want to study this in a rigorous way. And the studies of activating brown fat so far using cold have been somewhat limited, although you can take diabetic people and measure how much glucose they can kind of handle in an injection. So here's 8 diabetic people and how much glucose they can handle. Then you can ask them to sit around in shorts and a t-shirt at 57 degrees. So again, not even that bad for a couple hours a day, and then retest them. And each person will be able to handle more glucose. And this is good, right? This is an improvement in that diabetics person to ability to dispose of glucose. So activating your brown fat can work for you. So my lab focuses on 4 questions. We know that brown fat amount and activity is good. So we want to turn it on and we want to give people more of this. So to bridge that gap, we study how brown fat develops, and we use mice to do this for the most part because mice allow us all sorts of opportunities to manipulate things and look at whether or not they grow their brown fat. We want to know what happens when brown fat is activated. As I said, so far, the studies in humans don't really seem to support this idea that you can lose weight by activating your brown fat, But I showed you changes in insulin sensitivity, and we've studied, or I've studied, especially in my previous work at the Joslin Diabetes Center, changes in lipid profiles that occur when people are cold exposed, and potentially, other differences that would require the proper cold exposure regimen. Move to Maine, right? And then lastly, what we study a lot is, can we mimic the effects of cold on people using drugs? Because the fact of the matter is, even though I love Maine, people don't like being cold. They're not very fond of it. And if you give them the choice, they won't do it. And so what we want to know are there cold mimetics, and do these people who don't have brown fat need to just be supplemented with these cold mimetics? And so I'll just tell you quickly, what we do is we take, for example, blood from people who have worn the surgeon's cooling vest, and then we use a technique called mass spectrometry to measure hundreds of different molecules that are circulating in their blood to look for ones that change. And here's one that we're particularly fond of. It's called 12-13 dihydroxy 9-Z-octadecanoic acid, 12-13 dihome. And you can see in this one, this molecule about doubled in the circulation of all of the people that we had wear this surgeon's cooling vest. So what the heck is this thing? It is a derivative of linoleic acid. So it is linoleic acid you consume in your diet, and then through an enzymatic pathway, it's converted into this, this molecule with these two OH groups where a double bond used to be. And we can very accurately measure this in circulation using these deuterium substituted internal standards. And the research that I've done has shown that in mice, 12-13 dihome actually can recapitulate some of the effects of cold exposure. So it increases brown fat, fatty acid, and glucose uptake, like I showed you in the people who are wearing the vest. It can decrease circulating triglycerides in mice fed a high-fat diet. And then in people, we know that we've now measured, and others have also done large studies to support our findings, that 12-13 dihome is negatively correlated with BMI. So the more dihome you have, the lower your BMI, and it's positively correlated with cold-induced energy expenditure. And so this is really great. We can use it as a surrogate for those PET CT images. Those PET CT images are expensive, and also you have to give someone radioactive glucose. And so using this as a biomarker would be really valuable for studies that activate brown fat. So moving forward in my lab, we want to use 12-13 dihome to sort of ask whether or not 12-13 dihome can, first of all, regulate brown fat development and activity, and we have the mice to do that. That's how we do these things. But also what we want to do now that we have this biomarker for brown fat, we've identified people who don't have 12-13 dihome, and our sort of pipe dream is, can we treat these people to activate either brown fat development or activity in them? And of course, our understanding in mice, we want to identify new circulating targets that similar to 12-13 dihome. And this is research that still goes on at the Joslin and that we're working on here in Maine today. So thanks for listening. I hope I gave you a little taste of what we do in the lab. I am but a tiny cog in the wheel that does this research. I work with a fantastic team previously at the Joslin Diabetes Center and now here at MMCRI. And then, of course, both all across America and all over the world. And thanks, I also want to say thank you to my funding sources at the NIH. I put my email address and I would be happy to answer any questions that anybody had. Thanks for listening to my talk.
Thank you so much, Matt. As the last time we talked, I have a million questions, but I'll leave them for some other time. And just, Matt mentioned his email address. So if anybody's interested in connecting with our speakers after the coffee hour, we will be happy to provide their email addresses. I mean, that's part of why we're doing these events. And I know that a lot of different collaborations and just developing relationships have happened after coffee hours, which is a great side effect of these events. Okay, Matt, you can probably stop your share now. Thank you so much. And now we're moving to Kate Dickerson. Kate is a great friend of BioME's, always eager to help, and she's running the Maine Science Festival, which is coming to Bangor just in a couple of weeks, and BioME is a supporter of that event, so I wanted to just highlight it and show our community that this event is happening.
Thanks, Agnieszka. All right, hopefully I've done this right. And
We see it.
all right, great. I'm going to start it here. So I am thrilled to be here. Thanks for having me. I'm going to be a very different speaker than the first two and the last one, but the coolest part, as far as I'm concerned, is that exactly what those folks are talking about is what we want to highlight at the festival. So I'm just going to give you a very broad overview, hopefully a quick overview of the Science Festival, and then talk about this year in particular. The Science Festival started in 2015 after 3 years of planning. And I finally, after all this time, figured out the best way to explain it. It's like an art festival or a music festival, but it's all about science. So we run 70+ events and activities over the course of 5 days, where we are highlighting in any way, shape, or form that we can figure out how to do it, the science that happens in Maine and the people who are doing it. So you saw the previous slide was with Habib Dagher. He was talking about offshore wind in a session that we call 5-minute genius, where we give people 5 minutes to talk and then we cut them off with a cowbell. These are hands-on activities. I think this is from a field trip day. We run a field trip day for 7th and 8th grade students. It's the only part of the festival that's closed to the public, but what we're trying to do is show students where their education could go. We cover everything from the trades all the way up through postdoctoral research. One of the best things, as far as I'm concerned about the festival, is that we run events for all ages. So as you can see in this picture, you'll have everyone from a teenager to adults and people who may actually be retired, all in the same room learning the same thing. We also hold events in bars because, number one, that's a guaranteed way to ensure that people realize that we're for all ages, but also more importantly, it's a really relaxed atmosphere. So it gives the message that we are trying to give, which is that science isn't scary, science is some really cool stuff, come and hear about it. The other part, one of the secret sauce parts of the festival, as far as I'm concerned, are the sessions where we have talks or forums. And we ask our presenters to be as accessible as possible. So many of the people who present at the festival, it's interactive, right? They're going to talk, but it's not going to be a lecture like what you may have experienced in school. It's going to be trying things out. And I really like this picture because, well, first of all, it's kind of funny, but I also really like it because of what this session was. So, and this, I think, gives a really good example of how we approach science at the Maine Science Festival. We teamed up a vocal coach and a mechanical engineering professor at the University of Maine who studies the vocal cords. And they went through their different fields of study, so to speak. So the vocal coach talked about how she teaches her students how to use their voice in the best possible way. And, you know, one of the things she tells her students to do is sing through a straw. And she actually said she doesn't let her students sing a song until they can sing the whole thing through a straw. And then we went from that to the mechanical engineering professor, who then explained what was going on in your vocal cords as well, and then touching a little bit on your abdomen and your core about all the things so that you can protect your vocal cords by doing it this way. You learn how to use your body in the way that won't damage your voice. So it's just this really cool little insight into how applications of science often happen in places where you don't think about it. And that is in the arts. So one of the things since 2015 that has been vitally important for us is to make sure that arts organizations are included as part of the programming. Again, partly because it's cool and interesting and they are applying science all the time, but also because oftentimes scientists can learn from artists and arts and humanities folks how to communicate better. One of the other things we've done is dance. Physics and kinesiology of dance. These dancers were actually used as props, for lack of a better phrase, to explain why your body is able to move the way it does or why it's not able to move the way it does. We also do art exhibits. This one was an interesting exhibit it was curated by medical students at the University of New England. And it's from their anatomy lab. So what they did was they showed people kind of these beautiful artwork from photographs that they took of in their anatomy lab, but they would also explain to people how they use this to understand anatomy and part of their medical training. All of the art there was done on purpose to lead you to our big event this year. So we run 70 events and activities over 5 days. I run the whole festival is available to the public free of charge because we have supporters like BioME. But we do have an event. The only requirement of being at the festival is that you do your science in Maine. And the only exception is the headliner event where we bring in outside people. But this year, you can see the Maine 2020, the bicentennial. This was originally planned for 2020. The background is that the Gulf of Maine is warming faster than just about every body of water on Earth. And the Climate Change Institute at the University of Maine is among the world's leading researchers of climate change and what's happening. So when you combine that with the bicentennial of Maine, we thought, well, how can we use that to really accentuate science? So what I was led to was scientists have known about climate change for a really long time. That picture up in the left is Eunice Foote. She, in 1856, kind of hypothesized that carbon dioxide would warm things up and greenhouse gases. And then over there, the gentleman in the right-hand corner up here, that's Arrhenius. I can't pronounce his first name, so I'm not going to. He actually did experiments to show that if you added carbon dioxide to things, it'll warm up. And so then we come to 1938 with Guy Callender, who had the very first published paper showing that climate change is in fact warming up the atmosphere. So we have had all these, well over 100 years of scientists understanding that climate change is happening, and yet we're still not totally getting it until recently. So when you combine that with the Gulf of Maine warming faster and the Climate Change Institute, what you get, if you're me, is the warming sea. So I had been in conversations with a very long time with one of our arts partners, the Bangor Symphony Orchestra and Lucas Richmond. And I said to him, Lucas, why don't you write a piece? Why don't you write a symphonic piece about climate change in the Gulf of Maine? Because I'm not a composer. I figured, how hard could that be, right? Well, he needed a little help, which we knew. So to make sure that he could do it, we spent 4 months going around and having him meet with all of these people to talk about their work with climate change in Maine. They explained either how they're researching it or how they know it's impacting their community. And then we also went to middle schools around the state, where Lucas, we would talk about the project, and he would explain the music part. I or someone who was a scientist, who was one of these folks, would explain the science of climate change. And Lucas would ask the students, what do you want to hear from the piece? And to a person, the students said, some hope. I'm going to skip that. So what we have now is the warming sea. It's a symphonic exploration of hope in the face of the climate crisis. It's our headliner event for this year. It is the only, like I said, it's the only thing that we charge for. The tickets are actually, I'm hoping, relatively inexpensive. And it's not just the world premiere of that symphonic piece, although that is the full crux of it. What we're going to do is build up an entire evening around climate change in Maine. We're having an opening conversation with researchers who are working in the climate change world. And I actually just had it confirmed yesterday that Governor Mills will be joining us to talk a little bit about what Maine is doing with regards to climate change. We're going to show you Lucas Richmond's journey. So how do you take this renowned Grammy award-winning conductor and composer and teach him about climate change in 4 months so that he can actually write a piece about it? So we actually filmed every interview. So we're going to have a short documentary film. And then we're going to have the world premiere of the warming sea. And then at the end, because climate change is difficult. It's difficult to work through. It's difficult to understand. We're going to have a reaction from many of the people that Lucas spoke with, and we're going to send off the audience with a little bit of hope. What can we do, both individually and as a community, and a global world to address climate change. And one of the cooler parts of this, of all of these collaborations, I just wanted to throw this on here. Among the many people that I've had the great pleasure to work with include a lot of breweries. And Orono Brewing Company and Foundation Brewing Company did a joint collaborative beer to help us get the word out about climate change in Maine and this event in particular. And the beer is called Hope Begins with Truth, which is the concluding anthem of the piece. So that beer will be available next Friday, the 11th, all over Maine, and apparently a little piece of Massachusetts. So Matt, you can tell your friends there that they could look for that. And then finally, these are the rest of the events that we have going on. If you go to that website, you can see all of the different things. We are really excited to be back. We haven't held a festival since 2019. And I'm just over the moon to be able to share more about Maine science in a live setting with people. Thanks.
Thank you so much, Kate. This looks incredible. Obviously, BioME as a supporter, we've been sort of in touch with what's happening with the festival. I am so excited for this year. I think, you know, a lot of our members of our community are science geeks. So if you guys have kids or just want to check it out, once again, it starts March the 16th to March the 20th. Maine Science Festival website has all the details of the programming. And also I wanted to mention Kate has a podcast. Kate, can you just mention the name of your podcast? Because it's a really cool podcast.
I can, thank you. I actually had a slide, but I didn't want to go over. So the podcast is the Maine Science Podcast. Terribly original name, but the coolest part about it, I think, is that we do a deep dive and we just have conversations with scientists from all over the state to find out what makes them tick. And Agnieszka has been really helpful, and I use these coffee hours, maybe you don't even realize this, as a scouting tool oftentimes to figure out who we could talk to. But we've had everyone from people who are talking about quality control and beer to climate change to the Blue Shift folks and their space stuff. I mean, no science topic is too out there for us to talk about on the podcast. So thanks for that shout out.
Sure, absolutely. And if anybody's doing any cool work and you're interested, you could probably contact Kate and see if you're a good candidate as a speaker. Okay, so it is time for our last speaker today, and it's Dr. Anne Lichtenwalner, who actually appeared in Maine Science Festival in the past as a, I think, 5-minute genius. I may be wrong on this, so maybe Anne, you want to correct me if I'm wrong. And whenever you're ready, you can share your screen.
Okay, well, thank you so much. And I have to say that it's so exciting to be able to speak to you guys. I hope you can see my screen okay.
Yes.
Good. So I'm not going to be talking about an innovative teaching technology for cancer, nor am I going to talk about brown fat, nor am I going to talk about a really cool new beer and a symphony, but I am going to talk about the work that we do here at University of Maine in the Veterinary Diagnostic Lab. I've been here since 2008. And I am a veterinarian. I also have a PhD in reproductive physiology, and I've done a lot of work with animals pretty much my whole professional life, in the academic realm, in the clinical realm, doing some clinical teaching and service, and then also serving here at the University of Maine as the director of the lab. And I don't have a lot of slides today, but I'm very open to questions and comments. I want to shout out to this group, some of the members of this group, for being collaborators in the past, and also want to thank the Maine Technology Institute for helping fund this really great new laboratory that we have at 17 Godfrey Drive in Orono. This is our necropsy room. The former necropsy room in Hitchener Hall was a tiny little cramped space that wasn't as biosafe and biosecure as this is. And this is part of a much bigger building with a number of different labs in it that do molecular biology and techniques to help serve the Maine, really all Maine citizens who perhaps might encounter a tick on them. And we can basically tell you what species of tick and whether or not it's carrying some of the more common tick-borne diseases. We have a service that does some wonderful aquatic animal research support. We do a lot of contract work for vaccines and fish nutrition and general fish disease and health, investigations in our aquatic animal section, which Debbie Bouchard has led for many years, but it's a wonderful facility. We also have our new plant diagnostic lab. It's not totally new, but it's been kind of revitalized and reorganized by our Allison Smart, who is our plant pathologist. And then, of course, we have the insect folks that Jim Dill basically is our lead person on for integrated pest management. So my little team, I don't have a really big team. We would like to have more people. That's, I think, our big glaring need right now. But what we do is we do a lot of service to Maine's animal industries. And this can be a farmer that doesn't have a vet, has tried to find a vet and can't find one, needs help with an animal die-off, for instance, or it can be a veterinarian who might want someone to do necropsy on an animal that was lost, or to team up with them on diagnostic work to deal with a problem in their practice. So the smaller pictures show basically not just the big empty room, but what's you know, when it's busy. And in the middle, we have one of our student workers in the back there inspecting a number of cadavers we had to evaluate for a welfare investigation. And then our visiting pathologist, Manny Wenslow, who we have recently lost because she was hired away, which was great. That was, you know, we didn't have a full-time position for her, but we desperately need more expertise in this lab. We really need to staff a little more heavily. We have Brenda Kennedy Wade, wonderful technician who's been working with us for years. And so we can do animals from large to small. We've necropsied mice in this, we've necropsied chinchillas, we've necropsied, you know, many birds, mostly poultry. And then we have the occasional really large animal. So it's really nice to have a winch and the refrigeration capabilities to allow us to do that kind of work. We have, I also have about a 40% research and teaching appointment in the School of Food and Agriculture. And over the years, we've done a lot of really interesting projects that have to do with things that come into the lab. You know, we have, we get to get a feeling for, we sort of have our fingers on the pulse of what's going on with the animal industries in Maine because of the fact that farmers seek us out to help them with problems. A number of years ago, we had problems in the state with an infectious disease called Corynebacterium pseudotuberculosis, or CL. We did a bunch of work on that and did surveys for that in our region in both sheep and goats, developed a diagnostic test for it that we made available on a research basis in our lab for a number of years. We were able to give service, but to combine that with research productivity. And we were helped in that process by USDA SARE, a couple of grants, which we really appreciate. Currently, I have a grad student working on a really nasty parasite called meningeal worm. And it's one of those things that has a complex life cycle. But an intermediate host for this worm is, are both snails and slugs. The definitive host in which the worm reproduces is the white-tailed deer. And so you have sort of a mixture of risk factors that can occur on Maine farms. This parasite doesn't cause any problems to the white-tailed deer, but for small ruminants and for moose, and even other species of deer, such as our farmed deer, red deer, fallow deer, et cetera, it can be fatal. So it migrates from the GI tract of the affected animal that picks it up from grazing, for instance, and then it migrates into the central nervous system, and it can cause severe hind limb lameness and wobbliness, and then eventually proceeding to death as it migrates up into the brain. Nasty. So my student Rachel's work showed that on Maine farms, we basically worked with 6 small ruminant farms here in Maine, that about 4% of the snails that she gathered from those farms are carrying nematode larvae that morphologically appear to be P. tenuis. Right now, she's in the lab doing PCR to try to definitively show that they are p-tenuous and not anything else. We did undergraduate research projects where we looked at ducks as a way to mitigate snails on pastures for our small farmers. We found that ducks don't digest the snail. They eat them, they don't digest them fully, and then they pass them. So those ducks might be just slightly relocating the problem and not necessarily doing away with it. But mowing pastures in Rachel's work so far does appear to substantially reduce the snails on the pasture. So that's something farmers can use. We also, these sorts of projects that we do as undergraduate research projects, and then in this case, combined with a graduate project, can have unintended results that can be very interesting. We found that a lot of these snails were carrying microplastics. And so several students have worked on this and kind of done surveys and other work that help, I think, raise awareness of this issue on farms and document that it just does exist. We're not sure what it all means, but right now we've been working with farmer perceptions of what it means and maybe farmer awareness of where those microplastics might be coming from and how they might mitigate them on their farm. We basically always want to keep our ears open for areas of need for the Maine agricultural community. One that certainly came our way the other day, literally, was highly pathogenic avian influenza. So our lab, we always knew that probably our lab would be the first entity to detect it because of the fact that we do necropsies on hundreds of Maine birds every year that come from farms all over the place. So indeed that happened. And so because we are a lab that has close ties to the state vet, and of course we have to report any reportable disease, we were able to very swiftly get this sample basically validated as HPAI and let the state vet know so that they could start working with the USDA to control this nasty disease. So we felt good about doing that. We're sad it happened. We talked a little bit before about the sheep and goat disease. We try to stay strongly associated with the small ruminant communities, both because they're pretty vibrant, fun communities here in Maine. They do a lot of work. A lot of folks that we work with actually are academics themselves. Oftentimes, I always tell people when I speak to farmers in Maine, I have to really just sit down and think about the fact that most of them probably have an equivalent education or more education than I do. It's a great group to work with. Recently I was able to give a talk with, in conjunction with Robin Fowler-Pratt of the Northern Solstice Alpaca Farm about winter care of alpacas, and that was a pleasure. Also, we do service to our dairy farmers and we do mastitis prevention. We've developed some research projects out of that. And then finally, we're always looking for ways to improve our lab. We would like to, our biggest challenge right now is staffing. And that I think will, I hope, be mitigated over time, but we may have to get creative in terms of how we do that. I also want to make one more mention because this is a group that's biomedically very involved and well wired. We are working with Amplify Additive, a Maine company, to design and eventually market and produce biomedical implants that are orthopedic and utilize foam metals. And so that's been a really interesting project to be involved with over the years. With David Navant and Jim Weber and Ian Dickey. So I just wanted to mention that. And this is my dog Bernie, my very bouncy border collie. Who I have to say, I love her to death, but I recently sustained a broken leg because of my bouncy border collie. So if you do have dogs, especially COVID dogs, beware. Anyway. Okay, so that was really all I had to say, and I want to thank you so much for inviting me. And I have spoken at the Maine State Science Fair and Science Festival, excuse me, and it's a blast. great group of people to work with, and it's so fun to get updated on their activities. So thank you.
Perfect. Thank you so much, Anne, for highlighting the animal health research. You know, we have a lot of animal health companies that we're familiar with, but I think it's important to highlight the research that's done here, and maybe there are some collaboration opportunities as well. Okay, so now we have 10 minutes for Q&A. I do see some questions in the chat, so I will go straight from the top. The first question is for Theresa. Are these tools used in the physician's office, or are they given to patients to use?
They're used in the physician's office, so in a clinical setting. Yeah, we have toyed with the idea of providing the tool for patients and kind of gearing it toward their findings, but that's a bit more a larger project. And so the idea is get this out first and then see where it goes.
Perfect. Thank you so much. A question for Matt. Is it possible to convert white fat to brown fat at all? I heard about people taking dips in cold ocean and taking cold showers. Would those help with formation of brown fat?
Okay, yeah, so, and sorry, there was someone also, I want to say thanks for mentioning Daniel Levesque, and also this idea of hibernating animals. I want to say to anybody who's interested, Matt Andrews at the University of Nebraska studies these 13 striped ground squirrels, and actually they make lots of 12-13 dihome when they want to hibernate. And if you deprive them of linoleic acid, which is required for dihome, they actually won't hibernate. They just die in the freezing cold. So essential for that. The question about converting brown fat to white fat relates directly to our work on the development of brown fat. It is, and my answer, I'm ashamed to say, is it's complicated. It's definitely possible. Whether or not those cells can fully do everything that a real brown fat cell can do, people need to do more research on, and we're doing that. The polar bear swimming, I would say there's some really cool research from Dr. Michael Simons, that's S-Y-M-O-N-D-S, at the University of Nottingham, and James Law, who are really nice guys, who actually did exactly what you're talking about. They measured brown fat activity in these polar bear things. There's a really funny episode of Seinfeld about it too. But again, nobody's done a study where they ask, you know, what happens if you polar bear swim every day? You know, they've just said, well, does polar bear swimming activate your brown fat? And the answer is yes, and it does that mainly through your sympathetic nervous system. And I see one of my great friends, Dr. Christy Townsend at Ohio State University, actually in the audience. And I have to tell you, she would be an absolute expert on the communication between the brain and the nervous system and adipose tissue. As I mentioned, all those organ systems talk to one another. Okay, and then, yeah, go ahead. I know there's a third question. I wrote down all my answers. Sorry to jump the gun, Agnieszka.
Oh, no, that's okay. Hold on, let me find that question. Okay. Is there much known about the role of your molecule of interest outside of cold response and potentially brown fat activation? Is that the question you're referring to?
Yep, and yes, the answer is that there is. And what I want to be really clear on here is that I'm not selling anything, and most of the things that are known about 12-13 dihome outside of what I've studied would indicate that it's not really that great for you. So 12-13 dihome, as I said, is a derivative of linoleic acid, which is an omega-6 fatty acid, and a lot of people say omega-6 fatty acids are overconsumed by people, are not great for you. They are essential for life. You must consume them, but overconsumption is certainly associated with tissue inflammation. And so I would say that Dr. Bruce Hammock at UC Davis, University of California, Davis, is an expert in looking at 12-13 dihome as actually a pain marker that somehow 12-13 dihome signals to people that you feel pain, and that would suggest that blocking 12-13 dihome may treat inflammatory diseases, and Dr. Hammock has done a lot of really amazing research on that. I also want to point to my other colleague, Dr. Kristen Stanford, who's looked at 12-13 dihome as a function of physical exercise, and it also can be increased by physical exercise in addition to cold exposure. And then just because I saw someone tagging on in the end, I don't know if bears produce 12-13 dihome, and I'm not going to be the person to find that out, you know, for better or worse, but maybe volunteering.
Okay, thanks so much for covering all the questions. So this morning I saw a man jogging in the snow, not, in my opinion, wearing very warm clothes, and I thought he was crazy, but he wasn't. He was just producing brown fat. So maybe we should all try that. Okay, we do have a question for Dr. Lichtenwalner. No, hold on. Yes, I think so. Okay, what are the roadblocks for increasing your staffing at the veterinary diagnostic lab? You mentioned you had some problems with it. Are you having a hard time finding qualified people, or is it funding related?
Sure. What a great question. Kind of both, but more the first. Yeah, funding the kind of positions that we need is a challenge. So, and some of that is, it's a perceptual thing, so that it has to do with, you know, how perhaps we're marketing ourselves internally. I don't know. I do know that, you know, the services that we provide have been longstanding, and I think that they are appreciated by the agricultural community. But whether anybody's really seeing the need, we're out there pushing, trying to, you know, keep things going. Sometimes have to get very creative, have gotten very creative, but I don't think that that's sustainable. And I think we're going to need at least 2 or 3 people in this lab and in the fairly near future. So I am working with our extension leadership team. That I think is the most powerful place that I can work right now. But I think maybe need some help in, you know, kind of marketing that or whatever, just trying to get people on board. It's not my area. My area of expertise is more like, you know, with the microscope and, you know, in the necropsy room. So I'm not a natural at it. We will have, I have an immediate need for a technical person, just as a temporary hourly position. But if you have a talented person who's at the beginning of their career, maybe just finishing their student years and has some background in microbiology, please send them my way.
Absolutely. I mean, we don't, but maybe someone on the call does. One more question for you, Anne. Have you worked with any oyster farmers around Vibrio? Not sure what that is.
Yeah, Vibrio. So a nasty pathogen. And years ago, Debbie Bouchard and I collaborated on some shellfish projects. We found that it was difficult to get health surveys funded. So we wrote several different grants to try and do that. So the team of people who would be more appropriate to reach out to would be Debbie, and then like Heather Hamlin, I think she's done work in a number of different species. And she's another aquatic animal health person who's done a bunch of work in the past. I've collaborated with her on some lobster stuff. But I haven't worked directly with Vibrio things. If there's an emerging issue and there's a need, I'd be happy to help foster getting inquiries to the right people. Most of my work now has to do with avian and mammalian species. So, but thank you for the question.
Absolutely. Thank you so much. Okay, we are running out of time. I wanted to thank everybody for being with us today. Our next event is on March 25th. It's going to be our legislative briefing. Some of you may know that we educate the legislators on all things bioscience, and it's very important for members of our community to join us for that type of event. We'll be sending more information, but for now, if you want to put it on your calendar, March 25th at 9 a.m. Thank you again, and see you next time. Bye, everyone. Thank you to our speakers as well.